So, dear ladies and gentlemen, dear colleagues, dear friends, it's really a pleasure to welcome you to this week's or this month ESDR Kitchen. We have an absolute giant of dermatology and skin science with us. He's shaking his hand, but it's absolutely true. I don't think he would need any introduction at all. Martin Röcken is probably one of those few dermatologists who have had a huge impact both in clinical dermatology and skin science over the last decades. He actually had his training in Germany, medical training, and then dermatology training in Germany, Switzerland, including beautiful Lausanne, in Brussels. Then he moved on to do a postdoc in, again, Geneva and NIH in Bethesda, and came back then to Tübingen to head the department there of dermatology for more than 20 years. Martin Röcken has published more than 300 publications with a huge impact, including papers in immunity, science, Nature, New England Journal of Medicine, and so on. And even after his retirement a few years ago, he still continues to publish on such a high level. He has numerous awards and prizes. He has, I don't know, seven or eight honorary members, including obviously the ESDR, where he's also past president. To be honest, I don't know where to start and where to end. It's just a huge pleasure to have you here, Martin, and to talk about one of your really main topics that you covered over the last 20, 30 years. And that you also helped shaping. So it's really a pleasure to have you here. And Martin, I'll just hand over and give you the stage. Thanks for having accepted the invitation of Katia and myself to present here at this ESDR Kitchen.
Dear Katia, dear Curdin, thank you so much for the invitation, dear colleagues. It's a pleasure and honor to speak for the ESDR. There's still my heart. Over the past 20 years, I think there's one industrial invitation I took to speak, but I didn't accept any other. It was my own research there, but speaking for the ESDR, so it's an honor. Thank you for inviting me. The topic starts in Switzerland. May I have the first slide? About 20 years ago, Zinkernagel and Doherty got the Nobel Prize for their seminal discovery that T cells can kill target cells. At that time, they used virally infected cells. And what it shows and what was the important point there is that they do not kill any cell, but they kill only those cells with whom they can interact in a cognate way. That it means today we say MHC class 1 restricted fashion. And on the lower right, you see the panel number 2, and it shows that the syngenic mice, where they recognize MHC, there they kill most of the infected cells, while in strains that do not fit with the T cell, at that time it was not known the MHC, the T cell receptor was not that known. If you read on the left side, they say thymus-derived cells, immune cells called T lymphocytes. So these T lymphocytes that come from different mouse strains don't kill. Based on this, we have now tens of thousands, hundreds of thousands of paper on killing in viral diseases, in autoimmune diseases, and especially in cancer. But one point that I think is always neglected, you see it already in the first paper, in the first line, that the killing efficiency is 70%. And if you look to many papers, it's 80%, 90%, but rarely 100%. Then the question is tumor eradication with killing all there is? Next, please. So it's not the question to doubt it. I just took our own textbook, not to point to this book, but just to show that's common knowledge, and I believe also too, and I think it makes no sense to doubt it, that we have two major modes of killing that help us to fight against cancer and against viruses. That's the cytolysis with perforin and granzyme mainly. And on the other side, there is a Fas ligand interaction, which induces apoptosis. You see it on the right side, and there are now several other types of cell death. Next slide, please. So if we continue, then 10 years ago, until then, it was still a question of does it play in tumor immune control? And when the PD-1 antibodies came, Caroline Robert and her group showed 10 years ago also in Nature that tumors, at that time it was even not yet approved by the commissions, that anti-PD-1 antibodies induce regression of tumor metastasis is associated with an infiltration by CD8 positive T cells, while metastases that do not have this infiltration tend to grow despite the therapy. So again, we show that tumor cell destruction by CD8 T cells is highly relevant for humans. Just to tell, there's no doubt that we need this. Next, please. But the question I want to address is there also a role for MHC class II restricted interferon gamma producing CD4 T cells? So I will show some links that came up from the literature over the past 5 years, then go to autoimmune diseases, and then go back to cancer. Next slide, please. This is a paper published in Science almost 1.5 years ago, where they looked for the MHC, asked whether there is some MHC genotypes that protect against lung cancer. And much to the surprise of the authors coming mainly from UK and Scandinavian countries, they found, you see always upper line in red, green, and then blue, that in the red and the green line are those that are either smokers or past smokers. And if you look there, then we find whatever MHC is used, there's a difference between one type and another type. There's a clear risk groups that have lower risk than others. And what came out is that these 9 groups are not restricted by MHC class 1, which codes for CD8 T cell responder, but by MHC class 2. Though it's a MHC class 2 what codes for CD4 positive T cells that decides whether the risk of developing lung cancer is higher or lower. Next slide, please. And they even identified the peptide that is needed to protect from lung cancer. Next slide, please. And about 5 years ago, we and others showed in a series of papers that also the immune responses triggered by anti-PD-1 antibodies are associated or even depend on one side T cells, but on the other side on interferon gamma. And if there is no appropriate interferon response, then the tumors cannot really be treated. Next slide, please. So the question is, does it also relay, do we have experience with? Next slide, please. So what we showed that in the 90s, this is a famous paper in the Nature Medicine that was published by Jim Krieger, you all know, and they showed that if you killed CD8 T cells, then psoriasis improved. At that time, it was strictly believed that psoriasis depends on CD8 T cells. Next slide, please. And we started in a different way and showed that not CD8 T cells, but much to the surprise of us and others, was that T helper cells, which produce interferon gamma, and at that time we did not know IL-17, can induce a severe EAE encephalitis with paralysis. Next slide, please. And we can jump this. And if we treat these mice with interleukin-4, the Th1 cells are replaced by CD4 T cells that produce IL-4. We do not have a suppression of interferon gamma. We just induce IL-4. We have the same number of infiltrating cells, and the mice are protected from encephalitis. Starting from there, next slide, please. We asked whether this can also be translated to humans. Next slide, please. And 10 years later, we showed that if we treat humans, this was the first cytokine therapy for a human autoimmune disease, we treated psoriasis with interleukin-4, and what we see on the right side is that we have a dose-dependent improvement of psoriasis, and the psoriasis improves within 6 weeks by a PASI of 20 with the optimal dose. Next slide, please. And at that time, we also showed that's not interferon, but it's something else we did not yet know at the time. It was not yet published. And later we showed that's IL-17, IL-23, which correlates with the suppression of psoriasis. Next, please. So since the CD4 T cells are relevant for autoimmune diseases and some types of autoimmune diseases are destructive, we next asked whether these T cells could not also be used to treat cancer. Next slide, please. And the first did with an adoptively transferred tumor. It's a colon cancer, which was established about 20 years ago, and was transfected with EpCAM, which is an antigen which is frequently used or frequently found on tumor cells of the colon. And on the upper side, the slide, we see that there is no help with no treatment, and it's Th2 cells, and these Th2 cells, whether they're specific or not, simply no real. But when we treated the mice with interferon-producing EpCAM-specific T cells, CD4 T cells only, highly purified, then the mice were protected, and we did not observe over more than 100 days any disease. And then we asked whether it has to be antigen-specific or not. And on the lower slide, you see if there's any also controls that they don't produce interferons, they are not specific. Always mice die almost at the same time, but if we have Th1 cells specific for EpCAM and only these ones protect. Next slide, please. So this is still artificial and it's still used, but the big key question that brought us forward, took a long time because the experiments take years, was we got a model from Doug Hanahan. He is now working in Lausanne, and he developed a model of cancer which was used for something completely different already in 86, and these mice are transfected or are transgenic for large T antigen TAC. And on the right side, you see that the TAC, which is under the insulin promoter, is expressed by isolate cells. This is green, the TAC is red. And this T antigen, which we also find in Merkel cell carcinomas, which are also neuroendocrine cancers, induce the isolate cells to develop cancers over about between 10 to 12 weeks. Next slide, please. And the step, and what we then asked, if we treat these mice with CD4 T cells that are directed against T antigen, the beauty of the system, to my knowledge, the only one where we have CD4 T cells in transgenic mice that recognize a tumor-associated antigen, the TAC. So we made Th1 cells, treated the mice. Next slide, please. And what we found was the surprise is if you treat them with CD8 T cells, if nothing helps to make the mice living more than 20 weeks. But if we treated them with the Th1 cells, then the mice lived half a year when we stopped the experiment. And we expected that the T cells would destroy the islet cells and induce diabetes. But surprises did not happen. Instead, the mice did live, and as you see on the black dots, the blood sugar remained over the half a year almost normal. There was a small dip, is due to the mice that died. There were some mice who died, but more than half of the mice survived, as you see on the left side. Next slide, please. And then we looked for many factors, and this is one key slide there is that in the middle you see angiogenesis in the normal cancers. And if we treat the mice, they do not develop cancers. They have no angiogenesis. And then we asked whether this is a cytokine, and much to our surprise is a right column. When we blocked the interferon with an antibody or to knock out mice, then the tumors were not, this was not neutral, but when mice were treated and interferon was blocked, the tumors grew like hell, and the angiogenesis was terrific. You see on the right side, the little pancreas and the huge metastasis already in the organ. Next slide, please. So we concluded at that time that CD4 T cells can protect against cancer, can double lifetime, it requires interferon and TNF, it arrests the proliferation, and it's independent from CD8 T cells. Next slide, please. It depends the growth and the angiogenesis, and also it prevents the infiltration. So they asked what might be the mechanism. Next slide, please. So we had a journal club and looked between those that did grow on the left side and those that were arrested, and asked what is the beauty? Why are they more ordered on the right side? And that's now the last 7 to 8 minutes that I will speak about why the right side looks ordered. Next slide, please. So it took us the time, but what we realized is was a young medical student, colleague who unfortunately went into gynecology, but she found that the T cells are arrested. So if we take the T cells from the mice that were treated, they stopped growing even in the absence of any treatment, while the non-treated mice or tumor cells that were isolated from that did grow rapidly and exponentially. And on the left side, we see that it can be reproduced in vitro. If we expose the tumor cells to interferon and TNF, they simply stop growing, but they're still alive. And this can also be shown. Next slide, please. In vivo, we know that nevi express P16, which is the cell cycle arrest marker. And those tumor cells that were arrested in vivo expressed P16. They did not show the known Ki67 proliferation marker, and this is strictly dependent on interferon. Next slide, please. So what we concluded is that somehow interferon TNF activates the JAK-STAT pathway that then is known to stop to activate P16, to activate TP53 and P21, and to have a negative effect of MDM2 and the cell cycle proliferating CDK4 and CDK6. So they asked whether this is really true. So we explanted the cells. Next slide, please. And treated the mice with the classical checkpoint inhibitors. And on the left side, we see that the transplanted tumors did grow. They did not show the cell cycle arrest P16. They were full of Ki67. While when treated, they stopped growing. They were P16 positive, like nevi, and Ki67 negative. Next slide, please. So then we went on and asked whether this is really the interferon signaling, whether it's really the P16, and I showed just one single experiment from cell lines where we did knock out P16. Next slide, please. And whether we did treat the mice or not, the tumors did grow and were full of Ki67 and no P16. Next slide, please. So the question is this also true for the human situation? So we have a huge melanoma unit, and we had many patients that were treated, and as we know that half of the patients do not respond to the immune therapy, while about half of the patients have long-term remissions. So what we did is we took lymph nodes prior to therapy from those that did not respond and compared them to metastasis from those that did not respond, and compared them to metastasis that did respond. Next slide, please. And what we found is that with many aspects of genetics, they were similar, but on the right side, we see a difference. And what was different was losses of genes that were responsible for the cell cycle arrest, loss of P16, loss of the interferon signaling pathway, CHEK1, CHEK2. So if we had either loss of the interferon signaling or loss of a cell cycle arrest molecule or gains in drivers like CDK4, then the tumors did not respond to the immune therapy. Next slide, please. So this summarizes that in mice and humans, if there are loss in cell cycle arrest molecules, the tumors do not respond to immune therapy. And if there's a gain in either MDM2 or CDK4, that's the same. Next slide, please. So is this unique to us? And there was 3 years later a paper published in Nature Medicine, and they looked for tumors that did the metastasis that did outgrow after immune therapy. And in 8 or 7 of 10 tumors, they analyzed exactly these genes were either amplified, the proliferation genes, or deleted the cell cycle arrest or the interferon signaling molecules. Next slide, please. So if we summarize now again, on the right side, the ordered, on the left side, the chaotic tumors. Next slide, please. So it is really true that they are ordered, and this is again in this system. The beauty of the isolates is that they have very well expressed differentiation markers. It's insulin, the glucose receptor, and the glucose production. And what we see on the left side, the columns, is that these proteins are all expressed in normal isolates, while especially the insulin, which is the gray, and the glucose receptor, which is the white, are rapidly lost only by the tumor cells. And what happens if we treat them with Th1 cells? Next slide, please. Then the tumor cells are not only arrested, but as we see on the color slide on the left, they are highly differentiated. They express the synaptophysin, which tells us that the neuroendocrine cell, it expresses strongly insulin in the normal level, and they express the glucose receptor. So keep them highly differentiated. So since time is almost over, we jump the next slide. We jump over it. So I come to the next slide, please. So to summarize, we have shown most published, but part still on the way to be published, is that interferon TNF induce in cancer cells a cell cycle arrest. And this induction of the cell cycle arrest, this is the slides I did jump, depends on whether the genes that are necessary to arrest the cell cycle, the interferon signaling, the JAK signaling, or the TP53 are still present. If these are lost, then the cells can no longer be arrested. And if they are no longer arrested, then they lose the differentiation. They will lose their physiological function. That's a gain of genomic aberrations. So all this can be arrested by interferon TNF. The interferon TNF, by arresting the tumor cells, arrest the tumor infiltration, the tumor angiogenesis, and in data I did not show even the capacity to cause metastasis. Next slide, please. So to summarize, cytolysis and apoptosis by CD8 T cells or NK cells are key mechanisms. Also Th1 cells induce cytotoxic macrophages. But in addition, this is not enough. We think we can show that in addition, we need the arrest of those cells that do survive the killing. And this induction of the cell cycle arrest goes along with high differentiation and prevents the acquisition of genomic aberration. Next slide, please. So just the two last slides. This is a melanoma that has regression. And as you see on the left side, it's full of proliferating cells in red. And on the right side, the cell cycle arrest molecule P16 is lost, as we know from pathology. And if we now go to the regressing part, next slide, please. We see also in humans, melanoma cells, that the regressing parts, the melanocytes probably look very differentiated. They are not proliferating, but full of P16, which is the cell cycle arrest molecule. Next slide, please. So the conclusion is, besides cytolysis and apoptosis and other death mechanisms, we need a functional interferon signaling pathway in cancer cells. This is required to stably arrest the growth of the cells. It's needed to suppress CDK4/6, and it's needed to keep the cells differentiated and to prevent genomic aberrations. And the loss of cell cycle control mechanisms then is a marker for metastases that are resistant to therapies. I think this was the last slide, and this is a thank you to you all and all those who did the work. Thank you so much.
Thank you very much, Martin, for this really interesting talk. So exemplifying that there is still a lot more to know about how to treat cancer and all the mechanisms of metastasis. So please ask your question either in the chat or the Q&R, and so Curdin and myself will be more than happy to ask them to Martin, so no one can directly talk. So maybe I will start while people are writing with a first question. So did you look at these Th1 interferon gamma producing cells? Do they also secrete cytotoxic molecules, or is it like really senescence and no cytotoxicity?
Oh, yes. When we submitted its paper to Nature, we have 3 figures only to show that we did not find cytotoxicity. I know that you can induce cytotoxicity with CD4 T cells, but compared to what we have with CD8 cells, it's really minor.
Okay.
And if you have 1 million cancer cells, you kill, let's say, 100,000, but 90% survive. And the effect on is also to show that interferon TNF can also induce apoptosis, but this is really a cell cycle arrest, and the cells live. What we did, we took out the tumor cells and retransplanted them to normal mice, and they remained vital, but arrested for 3 months.
Okay. And so another question still on this like Th1 subset is, did you look at between the one that produce interferon gamma and the one who, well, I guess most of them do produce interferon gamma, but do you have like high levels of immune checkpoint receptors expression, for example, like PD1? When you have the anti-PD1, you will relieve the capacity to induce like melanoma cell senescence, for example?
I think we did not directly look on the PD1 of this on these Th1 cells, but what we did, we look where do they go, and you don't find them in the tumor. Most CD4 T cells are either in the draining lymph node or in peritumoral lymphoid structures. So probably most of the immune response happens not in the tumor, but around the tumor and in the tumor draining lymph node.
Okay.
And now we showed this at this cancer, but there's now a paper from, I forgot, in immunity that confirms this. That also in humans with anti-PD1, most immune responses probably happen in the draining lymph nodes and not in the tumor cell itself. There's also was a Nature paper 5 years ago, where they showed that there are paratumoral tertial lymphoid structures.
Okay. And so, sorry, Curdin, you want to
go ahead. You can follow up.
Okay.
I'll have afterwards.
Okay. Now, so after, I was wondering regarding like patients' treatment. So how would you foresee? So for patients basically that are resistant, you think that they are like, you need to target this like to promote this Th1 like interferon gamma response then, or it's both in responders and non-responders that you would need this response?
Exactly. I think we have, if we look to the data, we have about half of the patients who do not respond. And if they have a defect in interferon signaling or JAK2 signaling or a loss of P16 and amplification of MDM2, we have single patients who had these losses and who were negative for the BRAF mutation. And there we joined it with CDK4/6 inhibitors, palbociclib or so, and that had so many mutations but they had 1 year of healthy life, some of them. And I think we should see whether these patients do not respond better to we should exclude these ones from immune therapy and go directly, if possible, to CDK to inhibitors of BRAF/MEK early on.
Okay. So there is one question asking whether the tumor cells arrest in G1 or also G2 arrest in response to interferon gamma and TNF-alpha.
Yeah, you need always the two interferon TNF and it's a G1 arrest. We found some cells in G2 arrest, but we did not follow them further on. We focused on the role of the G1 arrest. G1.
Okay, there was a question from Julia Seneschal. from Bordeaux. I have a question. I mean, last year, I think, was the papers that came out in Science, 2 papers on combining JAK inhibitors with PD-1 immune therapies or checkpoint immune therapy. And it was in melanoma, and I think the other one was in lung cancer. And they said, like, if you go early on, JAK inhibitors. And they did add JAK inhibitors early on and said that if you have early inflammation, then you have regained the plasticity of the T cells again. And then you can render or switch non-responders into responders. So, I mean, looking at your data, that would be like a slippery slope, that you lose your effect that you show.
Yeah, I think this is a problematic situation because of the problem is everything can be toxic. And some patients who have or under PD-1 antibody, or if you have too much interferon and you exhaust everything, and it's like water. If you have too much water, you die. Yes, and there are some patients who have this overshooting interferon response, and there it's protective. But what is also published is that the JAK2 and 1 knockout tumors experimental and in humans, they do not respond at all to immunotherapy. And there are these patients who have immune-controlled cancers, and then they progress so fast that the patients die from it. So also the interferon TNF is a quantitative question. And this is, I read both papers. They also started to show that the JAK inhibitors indeed worsens the situation. They were sincere and had surprises. But the question is always the question, how much is too much?
And so this is really define a level where you actually add them, maybe even after a while, to regain function in the ones which do not respond.
Perhaps if you give it, and what is important in this paper, they give it only for a short period of time, I think, a week. And if you have a loss, it's permanent.
Yeah. So still a lot to do in that regard.
Yeah.
Any other questions? I don't see any.
No, I think.
Maybe just one follow-up question that I mean, you're still working forward. Just a little bit looking into into the future. Yeah. Do you think that the focus will be staying on T cells, or is it more focused now, switching to other directions that we have to combine it and have, target fibroblasts in the surrounding endothelial cells or so that have the microenvironment, or do you think that these combination therapies will just be for a subpopulation of patients?
Personally, if you go back now, the serious T cell investigation of T cells and immune response against cancer started in quite exactly in 1990, and more or less. And since we got stuck somehow, we had this jump up with the PD-1 antibodies. CTLA-4 makes 5%, but the breakthrough was, personally, I think that we somewhere reached the limitation of cytotoxicity. And the future, my personal belief, is to combine immune therapies with, for example, CDK4/6 inhibitors or something like this. There are some trials going on that were in the first glance not yet really successful, but they did not select for patients with, I think we have to make the genetic search for certain resistance parameters and then target these resistant parameters. For example, if the loss of CDK, increase of CDK4/6 or loss of P16, then give here CDK4/6 inhibitor like palbociclib and not do it in a broad way. I think in a broad way it doesn't bring so much.
Yeah. Thank you.
Thank you again, Martin, for sharing some of your ongoing results. So it was really nice, and for accepting our invitation. So before the end of this episode, so a reminder, so on November 12th, we will have the next sweet and sour episode. So be aware that the episode will be at 12:30 instead of 1:30. And so we will have Richard Weller and David Whitman for this episode. And so in December will be the second ASID episode, which will be on the environmental influences and dermatologic disease with Yumi Matsuoka Nakamura, and Ian Miles. So, and there, because of the time change, the meeting will be at 3:30. And lastly, so as a reminder for the people who could not attend, you can tell them that all the kitchen episodes will be available on the YouTube channel shortly after the meeting. Thank you very much, and hope to see you on November 12th.